Hello again. So did anyone go to a Zika endemic place? You did, I heard Puerto Rico, right? Just two.
Florida might be, I mean, it's my knowledge, who knows?
Yeah, in the US it hasn't spread among people. It's only come in. Anyway, so you'll be on the lookout. How many days did you return? Just yesterday, you two guys? So, you know, for the next week or two, you just watch out for fever, joint pains, and a rash. All right, the last time we met, we talked about intrinsic and innate defenses, and I want to wrap up immune defenses today with a consideration of adaptive immunity. So remember last time, intrinsic defenses are things that are always present in the uninfected cell. Processes like apoptosis, autophagy, RNA silencing, and a variety of antiviral proteins, a few of which we talked about. And then we talked about innate immunity, which is induced by infection, as opposed to intrinsic. And today we're going to talk about the adaptive system, which is tailored to the pathogen. And a unique feature of adaptive immunity compared to the other two defenses is that it has memory. It can remember the pathogen that's infected you. So let's take up at the end from last time where we were talking about how dendritic cells and other antigen-presenting cells, so dendritic cell is the main one we talked about, but other antigen-presenting cells like macrophages present peptides to the immune system. So here we have an epithelial monolayer. It's being infected by a virus. The virus is replicating and spreading, and these cells have sensed infection. They're producing cytokines like interferons, and those attract dendritic cells who sense them and become activated. These cells may also be dying and releasing viral proteins and nucleic acids, and again, the dendritic cell can take those up. The dendritic cell will then mature, it will become activated. It travels to the local lymph node where it will then present these peptides to the resident lymphocytes, B and T cells. And if they are detected as foreign, you will start to have an adaptive immune response, and that will consist of antibodies and a variety of cells that we're going to talk about today. First of all, let's talk about a little terminology so we're all on the same page. Lymphocytes and leukocytes. Leukocyte is a general name for white blood cells, and this includes all of the ones shown on this slide: monocytes, eosinophils, basophils, lymphocytes, and neutrophils. Leukocytes, white blood cells. Lymphocytes are simply a subset of leukocytes, and they include T cells, B cells, and NK cells, so you can see them up here. The lymphocytes are a subset of leukocytes, and these are distinguished by having variable antigen-detecting cell receptors. They can tell when another cell is presenting antigen to them, as you'll see today. So leukocytes and lymphocytes, I use the terms a lot, and again, when we're talking about lymphocytes, we're meaning a special class or subset of leukocytes. You should also appreciate where these cells come from. In the past, I think last time, I mentioned how when you have an infection and the infected cells are making cytokines, those cytokines get into your bloodstream, they go everywhere, they make you sleepy, they give you flu-like symptoms. They also get into your bone marrow and start the process of making more leukocytes that you need to fight off infection. So you don't carry all the leukocytes in your blood that you ever need. They have to come from somewhere, and they come from the bone marrow, where there are what we call multipotential hematopoietic stem cells. These are really amazing cells that can give rise to all of the cells shown on this slide, including leukocytes and the lymphocytes that are a subset. The multipotential hematopoietic stem cells in the bone marrow differentiate either into a myeloid progenitor or a lymphoid progenitor, and you can see the myeloid gives rise to thrombocytes, which you need for blood clotting, erythrocytes, red blood cells, and then a variety of leukocytes that we have talked about. and then macrophages and monocytes, whereas the lymphoid progenitor, as you might imagine, gives rise to the subset of leukocytes called lymphoid cells, including natural killer cells and T and B cells. So when I say the cytokines go in the bone marrow, this is what's happening to generate all of these wonderful cells that then come out and do their thing wherever the infection is. Back to the dendritic cell, on the left is an unactivated dendritic cell. It's near a mucosal surface. Of course, dendritic cells are not just at mucosal surfaces. I often use those as examples, but they patrol body cavities and other places in you as well. Let's say they are at a mucosal surface and there's some infected cells there. The cells, of course, are releasing cytokines, which are activating the dendritic cells. These dendritic cells have receptors for everything. Dead and dying cells being released at the mucosal layer can be picked up by the dendritic cell as well. And of course, viruses may also attach to toll-like receptors on the DCs, either on the surface or within endosomes. The result of all this is that the dendritic cells mature or become activated. And the activated dendritic cell is shown here on the right as the processes that give its name, the dendrites. It has MHC class II proteins on the surface, major histocompatibility locus class II proteins. These are important proteins for communication among cells. It also has a variety of other abilities, as we'll see. Now, what we're looking at here, this dendritic cell is displaying in MHC class II. The orange bar is meant to designate a virus peptide. It is a short amino acid sequence derived from a larger protein. And the DC has picked these up by endocytosis, for example. Here the peptides are in the endosome, and then they're brought to the surface of the DC during maturation. And this DC will go to the lymph node and present this to a T or a B cell and ask, is this foreign? Of course, it doesn't ask because it can't speak, but what it does is it simply presents it to the T or B cell, and during your development at a very young age, all the self-reacting B and T cells have been eliminated from you. So anything that reacts with a T or a B cell is deemed foreign by default. So here we see a peptide being presented to a CD4 positive T helper cell. So the CD4 lineage of T cells are called helper cells. But this DC could also present the peptide to a CD8 T cell or a B cell. The T cell will, if it recognizes as foreign, that peptide will become activated and eventually, in this case, differentiate into helper cells. Helping it along to do that are cytokines produced by the dendritic cell. So this is the bridge between the innate response and the adaptive response. The presentation of the peptide in the lymph node and figuring out whether that peptide is foreign or not. Now, if it's not foreign, if it's not a foreign peptide, in other words, if the T cell doesn't recognize it, then that's the end of the process. The dendritic cell will leave and go back to work. Now, here's an expanded view of what's going on in this presentation of the DC to the CD4 helper T cell. This is called the exogenous antigen presentation pathway because the DCs are taking up proteins that have been released, say, from dead and dying virus-infected cells. It's bringing these into its cytosol by the endocytic pathway, right? You see the early endosomes, the later endosomes being acidified as the pH drops. Now, if this were influenza, you should tell me immediately that flu is going to fuse with the membrane at low pH. But here there's no influenza virus. What's happening as the pH drops, there are proteases in the endosome that are activated, and they begin to chop up this protein into smaller peptides. And it's those peptides that are eventually going to be loaded onto the MHC class II complex, which you see here being made in the ER and shunted through the Golgi. Now, the cool thing here is that, there are lots of peptides everywhere in cells. So the MHC class II is made initially with what's called an invariant chain to plug the peptide binding pocket. So no other peptides will go in there. Eventually, when this MHC class II gets near the endosome pathway, the invariant chain is degraded. And then these vesicle-carrying MHC class II will fuse with the endosome, and those viral peptides will get loaded onto the MHC. They'll go up to the surface, and there they are displayed. So there is the MHC class II in blue. The viral peptide is in orange. And it is displaying, the DC is now displaying it, showing this peptide to a CD4 positive T lymphocyte. So that's where that peptide comes from. And you'll see later that peptides are also displayed on different kinds of MHC called class I to CD8 cells. These typically arise by infection. But this is the exogenous pathway. It is taking up proteins from the extracellular environment, displaying them in MHC class II to the CD4 positive helper T cell. All right, now, just like in every part of immunity, whether it be intrinsic, innate, or adaptive, viruses have antagonists. And just one example here for class II antagonists, human cytomegalovirus, a large DNA virus, which we'll hear about more later, interferes with the transcription of the gene encoding MHC class II. So what does that do? It prevents the display of the peptide on the cell surface, and so you don't get a robust immune response to this particular virus. As we will see later, the herpes viruses all have this property of causing persistent infections. They infect you when you're quite young, and they stay with you for your entire life. And one of the reasons they can do this is because they antagonize immune defenses at so many different levels. So again, just preventing class II from getting on the surface prevents the DCs from presenting antigen in the lymph nodes. Now, when you have an infection and these DCs go to the lymph node, you get proliferation of T and B cells. And the amount of proliferation is really quite interesting. And of course, you get proliferation if the peptide is detected as foreign, not if it is self. Typically, in your body, 1 in 10,000 to 1 in 100,000 of your circulating B or T cells will recognize any given antigen. But as soon as that cell recognizes the antigen presented by the DC, as we've just discussed, within 1 to 2 weeks, you get between 1,000 and a 50,000-fold amplification from one cell to between 10 and 50,000 cells. And this causes your lymph nodes to swell, which has a medical term, just like everything else, lymphadenopathy, swollen lymph nodes. And on the right is an image of a person with the lymph system indicated, and of course these are lymph vessels and capillaries throughout the body, and the little nodes you can see in various places. Some of these nodes are deep within you, but some of them are on the surface, close to the surface of your skin. Typical places under your neck right here, around your ears, under your armpits, and also in your groin. Please don't touch that right now. But when you get an infection, you can tell when you're sick because you get swollen glands. So if you got a rhinovirus infection, upper tract, you will often feel your lymph nodes swollen. And that is a telltale sign that your immune system is talking. The DCs have brought peptides into your lymph nodes. I find this really interesting. When you figure something out that's going on in your body, the next time it happens, you won't be scared. You'll think, this is just really neat. And so if you feel swollen lymph nodes under your chin here, in your jaw area, you've got some kind of infection in that area, because the DCs tend to go to local lymph nodes. You know, if you have an infection in your upper tract, the DCs are not going to go to the lymph nodes in your foot. They go to local lymph nodes, so you get a rapid response. That's lymphadenopathy. All right, the first question, which is a property of innate instruction of adaptive immunity? Presentation of viral peptides on MHC2 to CD4 T cells, endocytosis of viral proteins, activation of DCs by cytokines, sensing by toll-like receptors, or all of the above. 72% all of the above. Some of you just said A, which is certainly part of it, but it's not just A. Endocytosis of proteins, that's how the DCs take up the proteins in the exogenous pathway to present them, of course. Activation of DCs by cytokines is certainly right, and sensing by toll-like receptors. All right, so the DC has gone in the lymph node. The T or the B cell has recognized the peptide as foreign. You've had proliferation, lymphadenopathy. What's going on now? Here we have the effectors of the adaptive response made. The antibodies, shown here on the lower left as little red Y molecules, and the cell-mediated responses, the cells on the right. So again, we put an antigen of some kind into an animal. We can get the DCs bringing that antigen to the lymph node. One of the outputs is getting antibodies made. And here, the cells that the DC interacts with are B cells. And they have antigen receptors on their surfaces. And when they recognize a particular peptide in the DC as foreign, they begin to proliferate. They get activated. They become what we call plasma cells. And these plasma cells make a ton of antibody, like 2,000 molecules a second. They pour out antibodies, which are these red molecules, and those combine with the antigen. If it's a virus, it will combine with the virus. And the outcome of that we'll look at in a moment. So that's what we call the humoral response. You make these antibody molecules that move through your blood, move through your tissues, and bind the antigen for which they are specific. Now, the process of going from a B cell to a plasma cell is amazing. You initially select a single B cell that's specific for the antigen, and then while that's being amplified and you make antibody, you undergo somatic mutation. So you make more and more higher affinity antibodies, and all this happens in the lymph node until you get, after a couple of weeks, really, really high specific antibodies for the antigen. Now on the right, the cell-mediated response starts in the bone marrow with the precursors of the lymphocytes. They move to the thymus where they mature further, and then in the lymph node, we have two different kinds of T cells. We have the CD4 positive T cells, or T helper cells, and I showed you earlier how these can interact with a dendritic cell. When these are activated, when these recognize a peptide as foreign in a DC, they make cytokines. That's what their function is, these T helper cells. They make two kinds of cytokines, Th1 cytokines, which help cytotoxic T lymphocytes mature, and they make Th2 cytokines, which help B cells mature into plasma cells. So that's why they're called helper cells. They either help make antibodies or cytotoxic T lymphocytes. The other kind of T cell that's in the lymph node is a CD8 positive T cell or a cytotoxic T lymphocyte. The CTL is what the CD8 will become once it recognizes a foreign antigen and becomes activated. And as you'll see later, the job of the CTL is to kill a virus-infected cell, and we'll talk about how that happens later. So two different kinds of lymphocytes in the lymph nodes, and they give rise to two different functional classes, the helper cells, which make cytokines needed to mature either the antibody or the CTL response. And I've got a little dendritic cell here, just to remind you that all this gets going by an antigen-presenting cell, either a DC or a macrophage, for example, bringing antigen into the lymph node. Now let's talk a little bit about antibodies. Antibodies are Y-shaped molecules. They're shown on the left here. The little circular parts of these is called an immunoglobulin-like fold. It is a specific kind of protein fold that has since been found in other proteins as well. It's just shown schematically here. And as you can see, antibodies are made up of 4 polypeptide chains, 2 heavy chains, the longer ones, and 2 light chains. And the part of the antibody that binds to the antigen is up on the top here at the very tip. The hypervariable regions are the parts that interact with the antigen. And the top portion of this is called the FAB fragment. That's the part that binds antigens. And the bottom part is called the FC region. And this has other functions. It can bind to receptors on cells. So you can imagine a cell with an antibody bound by the FC portion. So that means the antibody combining sites are sticking up and it can grab antigen. And there are other interactions that occur via the FC portion. For example, complement can bind antibodies in this FC region, and all that can have effects on the immune reaction. So that's what an antibody looks like, roughly in schematic. It's not actually how it looks like in real life. On the right is the scheme of an antibody response. So in this experiment, we're injecting an animal. If you want to make antibodies, you have to put them in an animal. You have to put antigens in an animal. So we're putting antigen A into this animal at day 0. We're looking at the serum antibody titer. So we're taking blood at different weeks after injection, and we're asking, are there antibodies being made that can combine with the protein that we injected originally? So you see, after about 2 to 4 weeks, you begin to make antibody against A. It peaks at about 3 weeks or so, and then slowly declines. So this is characteristic of an antibody response. You get a peak synthesis and then declines, but you notice that the level to which it declines is higher than the baseline level of the naive animal at time 0. And so when you get a virus infection, you initially will make lots of antibodies, and they will decline in titer after a month or so, but you will always maintain a low titer, not 0, against that virus or the antigen. Now in this experiment, at 7 weeks, we then inject a mixture of antigen A, same antigen we used before, and a new antigen which is completely unrelated, different protein. We mix them both together, we inject them into the animal, and look at this, we get a secondary anti-A response, very rapid. We don't have a 2 to 3 week delay anymore, and it's more robust, higher titer antibodies. You can see that the peak of the red line is higher. We don't initially get an antibody response to the B antigen until 10 weeks or so. That's because it's the primary response against antigen B. So this illustrates primary and secondary antibody responses to different antigens. And what's going on here is memory. The animal retains memory to antigen A, and we'll talk about what exactly this is a bit later. Now, when you inject an antigen or a virus into an animal, there are different classes of antibodies that are made. 3 of them are shown on this slide, IgG in green, IgM in purple, and IgA in light blue. So here in this experiment, we've injected animals with poliovirus, and we're measuring at different days after immunization the titer of antibodies against polio in the serum, and moreover, we're looking at what kind of antibodies are being made. So you can see, if you look at the IgM plot in purple, IgM goes up initially very quickly. It's the first antibody to be made, and then it declines after 20 or 30 days or so, followed by a rise in IgG, which is shown in green. And finally, if we're looking at mucosal surfaces, if this is a pathogen that infects at a mucosal surface, you will often have IgA-produced secretory antibodies which come out of your blood onto your mucosal surfaces, and you see their uprise is a bit late. So what is this? What's going on here? Why do we have IgM and then IgG? Well, IgM is the first antibody that's made. It's typically a low affinity antibody, but IgM has the property of being able to form this pentamer of 5 antibody molecules joined together at their Fc, and that amplifies the affinity of the antibody for the virus or for the peptide or whatever the antigen is. So the affinity of an individual IgM is low, but when you multiply it by 5, you have a greater avidity for the antigen. Now, the reason this is done is because you can respond very quickly. To get a high titer or a high affinity antibody takes a long time of selection in the lymph node, and that's what's happening in the meanwhile with IgG. So IgM comes up first, high avidity, can take care of things while the lymph nodes are making high affinity IgG, and then those predominate later on. Now, when you are diagnosing virus infections, if one of you came back from spring break and developed Zika infection, they would take your blood and they would find that the Zika antibodies were IgM, which shows that you were recently infected. They will then look 2 months from now at what we call your convalescent serum. When you've recovered, you should have lots of IgG against the virus and very little IgM. So this is a very nice rule of thumb. IgM comes up first. Diagnostically, if you find a lot of IgM in someone, it means they're recently infected. If you find predominantly IgG, it means the infection was quite old. Now, secretory antibodies play an important role in defending us against infections at mucosal surfaces like the respiratory, alimentary tract, etc. And these are made just like other antibodies by plasma cells that are in lymph nodes, or in this case in the lamina propria, the area beneath the epithelial sheet. So this is a single epithelial cell. The lumen is up at the top, so this could be the respiratory epithelial lumen or the gut lumen, doesn't matter. Below that epithelial sheet, which continues on both sides, is the lamina propria, and down there are blood vessels and lymph vessels and cells like plasma cells. So if you have an infection in your epithelial sheet, the antibodies will be made by B cells, and some of them will be IgA. So IgM and IgG will typically stay in the circulation, and that doesn't do you very good if you have an infection going on at the mucosal surface. So some plasma cells make IgA, and this antibody has the specific property of being able to be taken up at the basolateral domain of these cells. They are made together with, at the bottom of the cell, there's an immunoglobulin receptor, and the IgA binds to that. It's taken into the cell by endocytosis, and these endosomes go all the way to the apical domain, and then the antibodies are released at the top. And the protein carrier that they used for this process is then cleaved off. So this is how you get secretory IgA. These are antibodies present at the mucosal surface, and obviously they're there to prevent incoming viruses from infecting. Now, when you inject an animal with a virus, you make lots of different kinds of antibodies to the virus. Viruses are complex proteins, and when you chop them up to make antibodies to individual peptides, you can get lots and lots of different specificities, or epitopes as we call them. Some of the antibodies we call are neutralizing antibodies. They will block infection with the virus. That's what we mean by neutralizing. Many of the antibodies will bind the virus, but they will not neutralize infection. We call those non-neutralizing antibodies. And as you'll see in a moment, the non-neutralizing ones can have a role in protection as well. But let's talk a little bit about neutralizing antibodies. Now, if you make an antibody that's neutralizing, it has the ability of blocking infection of a cell with that virus. Now, consider you have the first encounter with a virus. It takes you a week or two to make antibodies. Obviously, they're not going to prevent infection because you're already infected with the virus. They may help modulate the infection. But the next time you get an infection, you will have a memory response, which is very quick, and you'll make enough of that antibody specific for the virus to prevent infection. So the neutralizing antibodies comprise essential defenses, largely against second infections, not really the first one. These neutralizing antibodies can bind to virus particles and prevent them from infecting. That's exactly what we mean by neutralizing. And many of these are in the blood. IgG and IgM, of course, are circulating in the blood. They can neutralize virus there, or they can get into surrounding tissues. IgA has that special property of being able to get to mucosal surfaces, and that's a good place to be to block incoming viruses, of course. Now, in general, we say that antibodies can prevent virus infection, especially when it's a secondary infection after a memory response. But in certain cases, some antibodies are also important for recovery from virus infection. They're not as common as preventing infection, but they exist as well. So here is a very simple experiment that illustrates neutralizing antibodies. This is the plaque assay. Remember that a long time ago? We have a monolayer of cells, and on the bottom we have 4 plates with a number of plaques on each one. And the plates on the bottom, well, what we've got here is we have a series of dilutions of a neutralizing and a non-neutralizing antibody. So we've got 1 to 10 dilution, which is not very dilute, right? And you can see there are no plaques here. 1 to 100, still no plaques. 1 to 1,000 now, we've diluted out the antibody sufficiently so a few particles escape being bound, and we get 2 plaques. We keep diluting 1 to 1,000 and so forth. And you can see at 1 to 10,000, now there's no neutralizing activity. So this is what we mean by neutralizing antibodies. When you add serum, it binds the virus, prevents infection, and you can dilute it out. And typically, if you're looking for neutralizing activity, you do a series of dilutions like this so that you're convinced that you've got a neutralizing antibody. At the bottom is simply a non-neutralizing antibody, which at no dilution neutralizes infectivity. And these may bind the virus, but not in the right place to prevent infection. All right, so neutralizing antibody concept is really important. So here's an experiment to show you how if you have a good complement of antibody at the start of an infection, you can prevent disease. this is passive antibody protection against polio infection. So passive means you're injecting a preformed antibody preparation into an animal. And for example, you could immunize an animal with poliovirus and wait a couple of weeks and then take serum out, show that there are neutralizing antibodies in it, and then inject it into a second animal, which is what's shown here. So the readout here is paralysis. And on the y-axis, we have % paralyzed. And the control animal is inoculated with a certain amount of poliovirus. And after an incubation period, you score paralysis. You can see 100% of these animals are paralyzed. And now we are giving increasing amounts of antibody by simply injecting antibody into the animal. And then after we put antibody in, we inject the virus. So you can see the first column, it's highly diluted, so there's no protection. Less dilution at the third column here. You can see there's now 60% paralysis, so we've protected some of the animals. The column labeled 11, we've now protected a good fraction. This is more concentrated antibody. In the most concentrated preparation, all the animals are protected. So this illustrates the idea that antibodies protect you against infection. So how does an antibody neutralize infection? I know that's neutralize is a neutral word. It can mean many things, but in the context of virus infection, neutralizing means you prevent infection of the cell, and that can happen at several levels. All right, so here, for example, on the left, we're using rhinovirus to illustrate entry of a virus into cell. The rhinovirus is binding to a cell receptor. It's being taken into the cell by endocytosis, and then as the pH drops in the endosome, the RNA is released and infection begins. So now let's look at this process in the context of the presence of antibodies. So on the right side is what happens if we add antibodies to rhinovirus, and not just any antibody, but neutralizing antibodies. So one thing that can happen is that the antibodies can bind to the virus particle. You can see them illustrated here. They can bind to the particle and block attachment to the receptor. So the antibodies are sitting down on the particle exactly where the particle would sit on the receptor, so the virus cannot bind. That is easy to imagine, right? It's just a steric impairment. They can also cause aggregation of the virus. So for example, each antibody has 2 antibody combining sites at the tip of the molecule. Each of those ends of the Y can bind independently. So this antibody here, you can see, is binding 2 virus particles. And another one at the top is binding 2 more. So you can imagine many, many virus particles all bound in a big array. This we call aggregation. And that's not infectious because it's too big to get into the cell. Some antibodies will actually allow viruses to bind to cells. So here in the middle, we have an antibody bound. It hasn't blocked attachment to the receptor, but it's somehow blocking endocytosis, so infection stops at that early stage at the surface of the cell. And finally, there is a class of antibodies that can bind the virus. They allow binding to the receptor, they allow endocytosis, but uncoating is blocked. So the virus can get all the way down into the endosome where the pH is low. It does not uncoat because the antibody is present. Maybe the antibody is locking the virus so it can't open up as the pH drops and let the viral RNA out. Okay, so these are just a couple of the ways that we think that antibodies can block infectivity. Blocking attachment, causing aggregation, blocking endocytosis, blocking uncoating. I go through all of these because many people just think conceptually that a neutralizing antibody, which we say blocks infection, must act at the virus receptor stage. And certainly some of them do, but that's not the only way to become a neutralizing antibody. Now, antibodies can block infection in other ways that happen later than the attachment and entry steps of virus infection. So we just talked about ways that antibodies bind virus particles. But antibodies can also have an effect on the infected cell, and that's illustrated in this slide. So here on the top we have our virus. This looks like an adenovirus, and we have a neutralizing antibody bound to it. And in the previous slide we talked about all the ways that an antibody bound to a virus particle could block infectivity, and that's this line called neutralization here. On the bottom, though, is an additional way that antibodies can work. If this were an enveloped virus, this binding of antibody could lead to complement-mediated lysis of the viral membrane. So remember, complement is a set of serum proteins that can interact with antibody and lead to puncturing of membranes. So the complement system can recognize that this antibody is bound to an antigen on a membrane, and that triggers a series of events that will poke holes in the membrane, and that can also inactivate the viral infectivity. Once the virus has infected a cell, though, even after the infection has gone on for a while, you can also get killing of the cell by specific antibodies, and a number of these are shown on here. So here's an infected cell, and you may remember from our discussion of assembly, when enveloped viruses are made, one of the first things that happens is the viral glycoproteins are put in the plasma membrane, which then bud off to form particles. So here in this infected cell, we have a series of viral glycoproteins put in the plasma membrane. We're not budding yet, but you can see that if we have antibodies that interact with these glycoproteins, the antibodies will bind to that infected cell. And again, the presence of those antibodies bound to the cell surface will tell complement to go in and lyse the cells. And so this one here, Fc-mediated effector systems, complement binds the Fc region of the antibody right here, and it will cause lysis of this infected cell. And that will work for a neutralizing as well as a non-neutralizing antibody. Remember, these both are able to bind the virus particle. Both the neutralizing and the non-neutralizing antibodies can bind the virus particle. And here in this infected cell, all you need is antibody binding. It doesn't have to neutralize infectivity as it would on the virus particle. So either of these antibodies bound neutralizing or non-neutralizing can interact with complement. Then complement will lyse the infected cell and end the infection at that point. So the effect of an antibody can happen after the infection has begun. And there's some other mechanisms outlined here for how antibodies bound to these glycoproteins could inhibit infection. So we've talked about complement-mediated lysis. Some people believe that the binding of antibodies cause signal transduction pathways to occur through the glycoproteins that will inhibit virus replication. And it's also possible that antibodies bound to these glycoproteins will inhibit virus production. When these particles start to bud off, the presence of antibodies may inhibit that process, so maybe the virus particles are stuck on the cell surface. So the point here is that viral neutralizing and non-neutralizing antibodies can both impair virus infection in different ways. And I just wanted to show you this gorgeous picture, which happens to be poliovirus. It's a picture that I look at every day because it's the background for my cell phone. It's poliovirus bound to neutralizing antibodies, a single monoclonal neutralizing antibody. The antibodies are shown in purple. And it's just one kind of monoclonal, so it's binding one epitope. And this epitope happens to be located around the five-fold axis of symmetry. So there in the middle is a five-fold axis, and you can see 1, 2, 3, 4, 5 antibodies bound around it. And each five-fold axis is bound by 5 antibodies, so a total of 60 different antibodies bound. And this is an antibody that will block attachment. It's impairing the receptor from interacting with the receptor binding site. Do you think that viruses can evade antibodies, just as they've evaded all the other things I've told you about? Yes, for sure. Remember, I told you the correct answer is always yes. Evasion happens at every step. So here are 2 examples of evasion. So you inject animals with a virus. The host makes antibodies against the virus. On the left is rhinovirus. Rhinovirus's strategy is that it exists in over 150 serotypes. Now, a serotype is a virus that makes a certain antibody response that is specific for that serotype and not any of the other 149 other rhinovirus serotypes. So if you get infected with rhinovirus 20, you'll make a nice antibody response against 20, but the chances that you'll be infected in next year with 20 again are very low. You'll probably be infected with serotype 80, in which case the antibodies against 20 do you no good. So that's a strategy for evasion. And if you have 150 plus different serotypes that circulate globally, and you get one rhinovirus infection a year, you can do the math. You're going to get sick every year, most likely with a different serotype. So that's the strategy. On the right is the strategy of influenza virus. So this is the HA glycoprotein of the virus. And we're not showing the viral membrane here. Here, the membrane would be at the bottom. The top would be the globular head that binds sialic acid. Now, these colored regions are all epitopes. These are regions to which antibodies bind that are against the HA. The flu strategy is you get flu 1 year, you recover, you make antibodies, you have memory. The next year, you get infected again, but the strain has changed. It's made a 1 amino acid switch in any of these colored sites at the top. And now it's not neutralized as well by your antibodies that you made against the other strain. We call this antigenic drift. And that's why we often have to change the flu vaccine from year to year, because as the virus circulates in the human population, the pressure of antibodies on the virus selects for variants that have a slight difference and can escape neutralization. And we'll talk a little bit more about this when we talk about evolution later on. All right, our next question. Which statement about antiviral antibodies is incorrect? 1, they are important for protection against viral infections. 2, they only neutralize virus infectivity. 3, they may block virus attachment to cells. 4, they can be found at mucosal surfaces. 5, IgM is the first to appear, then IgG, which is wrong. Okay, most of you got B. They only neutralize virus infectivity, and that's the correct answer. They can neutralize virus infectivity, but as I showed you, they can have effects on the infected cell, which go beyond blocking infectivity of the virus. They can kill the infected cell, for example. Everything else is right. All right, so that is antibody immunity. We've talked just briefly about it, but it'll come back as we talk about viral diseases and vaccines. So you need to know the concepts. Let's talk about cell-mediated immunity. This, in contrast to antibody-mediated immunity, is essential for clearing most virus infections. And what's involved here are cytotoxic T lymphocytes, which arise from CD8 cells. And going back to our antigen-presenting cell, you remember how antigens were presented in the context of an MHC molecule. Now it's a slightly different scenario. We already have generated cytotoxic T lymphocytes in the lymph node, which are antigen-specific. They are now going to the area that's infected, and they're seeking infected cells to kill them. And these cytotoxic T lymphocytes make what we call an immunological synapse with an infected cell. So on the bottom is the infected cell, and all of our cells have MHC1 on them. And they display peptides. For example, if this was a virus-infected cell, it would be displaying viral peptides on its surface in the context of MHC1. Now, previously we talked about how dendritic cells display antigens in MHC2. MHC2 is restricted to very specific cells like antigen-presenting cells, dendritic cells, and monocytes. But all our other cells in the body have MHC1 so that they can show to a CTL whether they are infected or not. So the CTL comes up to a potentially infected cell, and it will recognize the peptide. If it's foreign, then the T cell will kill the infected cell. So this is called the immunological synapse. It's comprised of a variety of receptor-ligand interactions here. The basic point here is that the CTL is recognizing a foreign peptide in the context of MHC1, and that results in killing of the infected cell. So this is distinct from antibody protection because here you're actually killing the infected cell, not a virus particle. How does this work? This is called the endogenous antigen presentation pathway. So here we have on the bottom a virus-infected cell. And this cell is making viral proteins, right? Because viruses need to make proteins to make more of themselves. And what happens in a infected cell is that the cell is constantly sampling proteins in the cell and chopping them up and presenting them on the surface to make sure they're not foreign. This is a constant process in all of your cells. So here what happens is these viral proteins, they're tagged for destruction by the addition of ubiquitin. They're ubiquitinated. When you ubiquitinate a protein, that sends it to the garbage disposal of the cell, the proteasome. It's this big protease through which proteins are sent. They're chopped up into peptides. Many proteins are sent to the proteasome for various purposes. If you don't need them anymore, you can ubiquitinate them and send them to the proteasome. In this case, it happens to be a viral protein. It could be any cellular protein. Little peptides are made. They are then put into the endoplasmic reticulum by virtue of a pump called the TAP1 and TAP2 pump. TAP stands for transporter associated with antigen processing. It's essentially a channel through which the peptides go into the ER, and then they are loaded onto the class 1 molecule. The class 1 molecule, of course, is made by co-translational synthesis into the ER lumen. It's a transmembrane protein. It's then, the peptide is loaded onto the class 1 molecule. The class 1 is shipped up to the cell surface through the secretory pathway, and it's displayed. So now we have this peptide displayed on class 1 on the surface, and these cells are constantly under surveillance by cytotoxic T lymphocytes. So if this happens to be a virus-infected cell, on the top is our CTL. The CTL will recognize the peptide in the context of MHC1 by the T cell receptor, and that leads to lysis of the infected cell. If the peptide is not foreign, if it's self, no lysis will occur. So CTLs are recruited to an infected area by cytokines, for example. They will be sampling all the cells, and if they're uninfected cells, they will leave them alone. If they're virus-infected cells, they will be lysed by a property which I'm going to tell you in a moment. Now, of course, there are countermeasures to MHC1 as well, just like there was one that we mentioned for MHC2. Look at all the countermeasures for the MHC1 pathway. There are viral proteins that have been identified, and you don't have to know the individual proteins. I'm just putting them here just to show that there's an entity that's been discovered that interfere with the synthesis of the protein. They interfere with the transporter. They block the transporter so the peptide can't be put through into the ER. They interfere with the transport of MHC to the cell surface. Some of the viral proteins cause retention in the ER of the MHC1. Some viral proteins push MHC1 back into the cytoplasm. And some of them allow everything to occur. They allow MHC1 at the surface, but then they quickly cause it to be endocytosed. There are just so many mechanisms for antagonizing this among different viruses. You can see here we have lentiviruses like HIV. We have a lot of herpes viruses. These are all viruses that cause long-term infections. So they don't want to be cleared by the CTL response. That's why these antagonists have evolved. So this is a very effective pathway. Whenever you see so many targets for viral antagonism, you conclude this must be a really effective pathway for getting rid of infected cells. And in fact it is, and that's why viruses that want to stick around get rid of them. So lots of ways of countering MHC1. How does the cytotoxic T cell, or CTL as I'll call them, how does it lyse the cell? So here we have on the lower left, the CTL is recognizing an infected cell. The peptide is foreign. It's being presented by MHC1. The close apposition of the two cells leads to the insertion of a protein from the CTL into the cell membrane. That's shown on the right. It's called perforin. It's the purple protein here. And perforin works in one of two ways. It can make a pore in the target cell. Here we see lytic pore being made. That would be made in the infected cell, of course. So the CTL is depositing these perforin proteins into the membrane of the target. They make a pore. The contents of the cell leak out and the cell dies. So this is a very straightforward mechanism. On B is another mechanism. So again, the CTL makes a pore in the target cell membrane using these perforin proteins. And then it inserts a protein called granzyme B. Granzyme B goes through the pore into the cell, activates programmed cell death, and that's how it kills the cell. So two distinct ways of killing. One is by apoptosis, and one is just by having all the contents leak out of a pore. So that's how a CTL kills a virus-infected cell. So here's an experiment where you can see how the production of CTLs correlates with a virus infection. So on the top here we have infected an animal, and at different days after infection, on the top panel we measure virus titer. Here we're looking at the spleen. We're looking at log virus titer per spleen, and you can see after inoculation on day 0, the virus titer in the spleen goes up really quickly in a couple of days, and then by day 15 or so, the infection has been cleared in this animal model. On the bottom is a graph of the induction of CD8 CTLs. And so we have two different kinds of information in this graph. In the green bars are all of the CD8s that are being generated in this virus-infected animal. So you can see by about day 6 or so, they start to rise. They peak at day 10 or so. So that's slightly after the virus titer begins to decline. And in fact, what's happening here is the virus titer is rising, and at the same time, the CTL titer is rising. It's clearing the infection, so eventually, virus titer comes down. The blue the blue graph, or the blue line on the bottom graph, are what we call epitope-specific. We actually ask, what are the CTLs that are made which are specific for this particular virus epitope? And you can see those go up as well by day 10, but they persist. So the epitope-specific CTLs stay for a long time. There's memory. You don't need all these other CD8s around. You just need the ones that are specific for the virus. But this is a nice example of how there's a correlation between clearance and the induction of virus-specific CTLs. How do you tell if a particular virus infection is cleared or improved by antibody versus CTL? So here is an experiment that addresses that. You know how much I like experiments on exams, right? So here's an example of that. Here we're asking, what's more important in protecting animals against monkeypox virus infection? We have 3 different manipulations. So on the top line, so we're going to vaccinate these animals on day 0. And in the top line, there's no manipulation. We're going to leave them alone. We measure neutralizing antibody on day 22 to the virus. So you can see here, by 3 weeks, we have a nice neutralizing antibody response to the virus. And then we infect the animals. We challenge them with virus on day 28. And we measure fatality. That's the outcome or the readout of this experiment. So here, all the animals are protected by this vaccination. Very good. The next line, we vaccinate day 0. We then deplete the B cells from these animals. We can do that with an antibody. And then we measure neutralizing antibodies. There aren't any. No surprise, right? You take out the B cells, you're not going to get neutralizing antibodies. We then infect these animals, same day, 3 out of 4 are dead. And they have very low neutralizing antibodies. So the conclusion is, looks like antibodies are really important for protection. Now you're probably thinking, what happened to that other animal? And this is the vagary of animal experiments. Random things always happen that you can't explain. All right? So we have no idea why this animal survived. Certainly not because they had neutralizing antibodies. And that's the way animal experiments are. Last experiment, we give them a vaccination. We deplete their CD8 cells. Instead of B cells, we take out the CD8 cells. No effect on antibody production. Day 28, we infect them. They all live. You don't need CD8 cells for protection. In this case, you can see clearly that you need antibodies. You can imagine another scenario where maybe CD8s are needed for protection, and we do know some examples of that. Okay, so when you are infected with a virus, we've talked about antibody responses, we've talked about cellular responses, and I keep telling you antibodies are important for protection and CTLs are important for recovery. I haven't showed you data about that, but that's what we generally think. But again, there are exceptions. In biology, there's always exceptions. Sometimes antibodies are needed for recovery and vice versa. So how does the immune system know what kind of immune response to make against a particular virus? It doesn't make all kinds of responses. That would be a waste of resources. So how is the correct response made? It all starts in the lymph node. When the dendritic cell that's picked up antigen comes into the lymph node and starts to interact with B and T cells, very early on, that is where the decision is made whether to favor an antibody or a CTL response. So how does that work? Well, that involves the two different classes of CD4 cells that I mentioned earlier, the Th1 and the Th2 cells. Now remember, in the lymph nodes, the DCs and the macrophages presenting antigen, they come in, they present these antigens to the T helper cells or the CD4 cells. And at that point, what the two cell types exchange determines whether these cells are going to become Th1 or Th2 cells. So that's cartooned on the lower left. So in the lymph node, we have our antigen-presenting cell interacting with an immature CD4 positive T helper cell. And depending on the nature of the antigen and the kind of cytokines that the antigen-presenting cell is making, this cell can either become a Th1 CD4 cell or a Th2 CD4 cell. Remember, a Th1 CD4 cell will make cytokines that lead to the production of CTLs, cytotoxic T lymphocytes. The Th2 class of cells make cytokines that allow B cells to mature into plasma cells. So Th1 is a cytotoxic response, Th2 is an antibody response. So here on the right is a cartoon of that. We have our antigen-presenting cell, which has come into the lymph node, and it's presenting antigen. And depending on the nature of the antigen, so depending on what toll-like receptor it has interacted with, or cytoplasmic helicase, the nature of the cytokines that are produced, it can either cause differentiation of the CD4 into a Th1 or a Th2 response. Now, in general, as I said, Th1 are cytotoxic T lymphocyte responses. These typically occur when we have a virus infection, when you have a tumor. Any intracellular pathogen typically favors the Th1 response. Th2 responses, which make antibodies, extracellular pathogens, so a virus is also an extracellular pathogen, parasites as well. So it is very specific for the particular pathogen, and beyond that, even for the particular virus. So when you want to make a vaccine, for example, you need to know what is important for protection, whether it be antibodies or CTLs, and you have to make your vaccine tailored in a way that will make the right Th1, Th2 response. So this happens very early, and this determines how the antibody response evolves. The immune response, I should say, evolves. So the next time you have a swollen lymph node, just think of all the stuff that is going on in there, and you have a better appreciation. All right, our last question. For some infections, CTLs are more important for protection than antibody. How is the CTL-antibody balance determined? By toll-like receptors, by intrinsic defenses, by autophagy of infected cells, by the mix of peptides and cytokines presented by DCs, or it depends on whether the capsid is icosahedral or helical. Mix of peptides and cytokines, that's it. A few for the others, but the others are technically okay, but the best answer is D, because ultimately all the other stuff leads to the mix of peptides and cytokines that are presented by DCs. Now I want to go back to this slide just for one moment. Now I don't want you to think that this is a digital or a binary decision, that you get either Th1 or Th2. It's typically a mixture. One will predominate over the other, but you never have exclusively one or the other. I don't mean to give you that impression. There is also cross-regulation. For example, when the response biases towards a Th1 response, the cytokines that are made suppress the other response. So Th1 cytokines, which are needed for CTL maturation, will then suppress the Th2 response. So it will enhance Th1. And the opposite is true. If you make cytokines to enhance the Th2 response, they will suppress the Th1 response as well. All right, let's talk about memory. Adaptive responses provide memory. If you get infected a second time by the same or a similar pathogen, you'll get a more rapid and specific response. It takes a lot less time as compared to the first response. And this is what distinguishes the adaptive response from the innate response. Innate responses don't have memory. And this is the basis for vaccines, the fact that we do have memory. If we didn't have immune memory, we wouldn't have vaccines. There wouldn't be any point. So let's explore memory just a bit. Here's one of the first experiments to show that there is something called immune memory. And this was done long before we knew what a virus was. We knew that there were infectious diseases, and there were epidemiologists who studied them. And with this particular case, there was an epidemiologist who was studying measles. And in he actually did this in retrospect because he didn't live this entire period. He looked from historical records. There was an outbreak of measles on the Faroe Islands in 1781. So you can see they're between Iceland and the UK. Back then, relatively isolated, and not like today where a plane is probably landing every day and bringing lots of people in. Maybe you'd be lucky to have a boat come by once a year. So you're pretty much alone for a long time. Outbreak of measles, for the next 65 years, no measles. Why? No one was bringing the measles onto the island. And after that first outbreak, population gets immunized, nowhere for the virus to go. In 1846, there was another outbreak, and all the people who survived that first one, none of them were infected. They had immune memory. So this shows that immune memory can last a long time, at least 65 years, and you don't need to have the virus around to maintain it. So some people used to think that you need to have antigen present tickling the B cells constantly to keep them at a low state, but that's not true. Immune memory lasts because the B cells remain in you for a long period of time, and they're self-renewing. So what exactly is this? So here's an experiment to show immunological memory. We infect an animal right here on the left, and we measure either antibody or T cells. We can have antibody or B cell memory. We can have T cell memory. They're separate and distinct entities. Between 7 and 14 days, we have an immune response, again, antibodies or T cells, that wanes, as I showed you earlier. And then maybe 2 years later, in this example, you get another infection with the same agent. You have a very rapid increase in the immune response, and it's of a greater extent than the original one. So that's a feature of memory. It's very fast, and it's very specific. And because of this, you have a mild or maybe no infection whatsoever. So here we have a mild or inapparent infection as a consequence of this very rapid response. And initially, you have your first infection in a couple of weeks, you get sick, hopefully you live, but your immune response isn't quick enough to prevent infection. Now, of course, most infections are prevented, right, by your intrinsic and innate defenses. These are only the ones that get past those and encounter the adaptive response. That's the last chance we have to clear an infection, antibodies and T cells. And so you get a little sick, but eventually they kick in and hopefully they help you. So memory consists of what we call memory B cells. So these are B cells that have been selected in the lymph node to be antigen specific. Some of them have gone on to proliferate to plasma cells, but a pool of B cells specific for that antigen remain in the spleen and the lymph nodes, and they don't make antibody unless antigen comes back, unless you get another infection. Then those B cells will be found by DCs, and they will rapidly expand because you don't have to go through any of the selection and hypermutation that you would normally do in the first round in the lymph node. We think also, in addition to memory B cells, there are plasma cells that live for a long time. These are already antigen specific. They've turned off their secretion of antibodies, but they live in the bone marrow, and again, they can come out and produce antibody when they are needed. So that's the basis, in a simple, quick way, that's the basis of immunological memory. Now, some viruses and other microbes are better at making memory than others. For some viruses, for example, the memory is very poor, and we don't understand this at all because the basis of memory is poorly understood as a whole. And as I said, there are also memory T cells, so you have both antibody and CTL bases for memory. Now let's close up this discussion by just looking at two areas of the body where virus infections occur and try and put together all of the defenses that we've talked about. So here is the skin. So we have, in much of our body, as you know, we have immune systems to protect us. The skin is not without exception. Of course, the very upper layers are dead, but below it, the epidermis is full of cells of all sorts and blood vessels and so forth. And then here is the endothelium, and there's a blood vessel down here at the bottom. And so the skin has lots and lots of cells that participate in immune defenses, as you might imagine, because it's right there and it's easy to be attacked. So the skin, the bulk of the epidermis is made up of keratinocytes. These are what make up your epidermis, and these can produce both class 1 and class 2 molecules. So if they are infected, they will display peptides on their surface. They will make cytokines of various types that will bring in dendritic cells, which are constantly patrolling your skin. The ones in your skin are called mobile Langerhans dendritic cells. And again, they patrol looking for foreign antigens, and they can be attracted by an infected keratinocyte. They will then become activated, carry the peptide to the lymph node, and we've talked about what goes on there. And this is the bloodstream at the bottom here. So then the T cells and the B cells that are amplified in your lymph nodes will get back into the circulation, and they will home in to this infected area by virtue of cytokines that are being produced by the infected cells. So here we have Th1 and Th2 cells going into the skin and elaborating products, cytokines, which are going to help make either antibodies or cytotoxic T cells. So this is what's going on in your skin, perhaps when a mosquito bites you and deposits some virus there, and it starts replicating in keratinocytes. Now let's look at the gut. The gut has differences, of course, a lot of the same principles. Here at the very top, the lumen of the intestine, and of course there's a sheet of epithelium that we've talked about, which is your barrier between the intestine and the lamina propria, the underlying tissues. And these epithelial cells, of course, can be infected, and when they are, they will attract antigen-presenting cells like DCs and dendritic cells, and they will bring antigen via the blood or lymphatics to the lymph nodes. The T cells and the B cells will come back via the circulation and go into this lamina propria. The gut is unusual in that it has its own sort of lymph nodes that are called Peyer's patches. These are collections of lymphoid cells where some interactions of dendritic cells with B and T cells can occur, and those come right out and can then do their thing in the underlying area. So if there is ever a infection, those are the defenses that take care of it. And you can see the M cells are sampling antigen in your lumen, so if this is a virus, they will sample it, and if it's foreign, the DCs will find out, and in will come B and T cells. One last thing that's very interesting that's not shown on this slide, there are a variety of cells interspersed throughout this area. They're like dendritic cells, but they don't move. They have processes that go through the epithelium, and they wave in the intestinal lumen, and they grab antigens and put them onto the lower side where they can interact with some of these B and T cells that are resident in this area. So it's a very unique way of sampling the contents of the gut, whether it be a virus or just an antigen. This all sounds really nice, that it helps defend you and fight off infections, and you may think it's great, but we're going to see next time how this defense system that we've talked about for 2 days, while it can clear many infections, it can also be your worst enemy.